Okay, so we're going to finish talking about cell division. In the last video, we talked about how cells are living, so they have a life cycle. G1 is like the early stage. As soon as new cells are created, they enter, so this is like the end of creation, and then they enter this G1 phase, which is sometimes it's kind of the whole life cycle of the cell. They can enter what we call G0, where they don't move any further in interphase and they stay there. But G1 is where a typical cell, like a skin cell or something like that, is just doing work until it's triggered in some way to create or synthesize more DNA. So S phase is where they make more DNA, and then it just goes back on into G2 phase, where it's just getting ready to reproduce. It's getting ready by making the rest of the cell kind of sedated, so it kind of stops being as busy as it was, and maybe even replicating some of its organelles so that there's plenty for both new cells to have. And then it enters what we call the M phase, and that could be mitosis or meiosis. And so for right now, until we get to the reproductive system, we're going to talk about mitosis. So if you'll flip this paper over so the holes are down toward you, we're going to make kind of just a whole diagram of prophase, which is the first and longest phase of mitosis. So I'm trying to number my pages here, especially making these videos. I kind of get confused on which page goes where sometimes. So this is BR4, so Biology Review 4. Prophase, pro means before, so this one happens before any other phase happens. And a lot of things has to happen. Now remember, in this picture right here, we're looking at an entire cell. So here's the nucleus where the DNA is winding up into chromosomes. We could draw the ER and the Golgi apparatus and the mitochondria and the ribosomes and all of the little vesicles and all of the fibers that hold the cell out. We could draw all that stuff that goes in here, but then that's, kind of hard to see what we're looking at. So this isn't just an empty, quiet space. You have to remember that this is full of stuff that's very busy, makes the cell do its work and stuff. So when the nucleus has to dissolve or disappear during this phase, the brain, I guess you could say the brain of the cell, where the DNA is exposed. Well, if this is damaged, the cell no longer has the correct instructions. It can't make, it can't do its job correctly. So it's very important for the cell to chill before this is exposed. So a lot of things have to take place. The loose DNA winds up into chromosomes. The nucleus begins to dissolve so that then these spindle fibers that are going to come out of these centrioles can get to those to pull them apart. And then all the cell work and workers must basically stop or, go to sleep or, chill out or be sedated or whatever you want to say, so that the activities slow down enough for this not to get damaged when it's exposed. And then the centrioles migrate. They move to opposite sides or poles of the cell, so they just kind of move across from each other on opposite ends of the cell. And that's I just draw it like a little squiggly line, and then these spindle fibers are coming out. And I put these arrows just to show that this one's going that way and that one's going that way. So that by the end of prophase is so long that sometimes you will see a pro-metaphase where they've combined it with the next phase. It's like this is kind of in between the beginning of prophase and actual metaphase. But prophase is so long that by the end of it, there needs to be no nucleus. The DNA is in full chromosome form. And, there's a couple of reasons why DNA needs to wind up into chromosomes. Number one is, and we talked about this in biology, but number one is that if you will remember that the cell itself is microscopic cell. So, you're made of trillions of these. Each one of them has a tiny nucleus inside, and inside that nucleus is like 8 to 9 feet of DNA. And by the time it's replicated, because remember, right now we're right here. Well, we've already replicated that 9 feet of DNA, so now we probably have like 18 feet, possibly, of DNA inside this extremely microscopic nucleus. So that's a lot of DNA. So it needs to wind up, because when it's being pulled from one end of the cell to the next, to the other end of the cell, it doesn't need to be like single strand pulled. It's a lot faster if you have this bundle and it's just pulled. So it's faster. It's safer for the DNA. If I gave you yarn, I gave you this example in biology. If I gave you yarn and said, hey, take this home and make me a blanket out of this, and you took it by one end and you just like walked home with it, it's going to be damaged by the time you get to the house. It's going to take you longer to get it there, and it's going to be damaged. Same thing with DNA. It doesn't need to be pulled single strand. It's going to take forever for 9 feet to go from one side to the other, and it's much better chance of being damaged. So it winds up into this tiny, nice little package so that it can be moved faster, easier, and more safely for the DNA. So there's no nucleus protecting the DNA. It's in full chromosome form, and the cell has kind of like chilled out all of its work. Okay, so that is prophase. The next phase, if you'll just remember PMAT, you'll remember them in order. So the next phase is metaphase, and meta doesn't necessarily mean this, but meta middle, just the way I remember, that this is the one where they line up in the middle. The centrioles have reached opposite ends of the cell, and they've produced these spindle fibers. And these fibers have I think about like a little microscopic outstretched slinky. They have this potential energy of pulling back this way. So something that I did forget, I'm sorry to do this, but I can't restart this whole video, is when you're drawing your chromosomes, it needs to be 3 different colors. So whatever 3 colors you used on this picture, I want you to continue with those 3 colors throughout. So what happened is when this replicated, let's say that, let me find a piece of paper that's blank, or almost blank. Let's say that these chromosomes were the ones I got this, let's say this is chromosome number 1. I got this chromosome from my mom and this one from my dad. And this is chromosome number 2. And I got this one from my mom and this one from my dad. And this is chromosome number 3. And I got this one from my mom and this one from my dad. During the S phase, all of those replicated. So now I've got like 2 copies of my mom's chromosome number 1, 2 copies of my dad's chromosome number 1. Two copies of her number 2, 2 copies of his number 2, and so on. You do that for all 23 pair. We don't draw all 23 because that's just confusing. So all 23 pair do this, but we're just going to draw 3 of these homologous pair of chromosomes. So they've all replicated, and these spindle fibers have come to join them on what's called the centromere. And the centromere is like just a super tightly wound up area of the DNA. Is very tightly wound up there on the chromosome, so that, if you think about it, if you pull on something that's really weak, you're going to snap it off. So this is really tightly wound, and so when this pulls, it doesn't snap it off, it's able to pull it. So the centrioles and the spindle fibers are also putting a pressure like out here. This cell membrane is very flexible. That's why, when I do this, you don't hear a bunch of cracking noises, because my cell membranes are movable. Unlike a plant, their cell membranes are too, but they have those cell walls around it. So we don't have those cell walls, so these are very flexible, and these are being pushed on. So the cell is elongating, and we'll draw that here in just a minute. And so it's stretching out the cell. And so I kind of think of a piece of pizza dough. And as you have a ball of dough, and as you slowly pull it, it just elongates. And then eventually it's going to pinch off in the middle. And so that's kind of where we're going with this. Okay, so the next one is anaphase. So those centrioles keep pushing out on the outside, and it's elongated this cell. Just think ana away. Again, that's not necessarily what that means, but just kind of help you remember that. But these chromatids, so this was my mom's number 1, and this was her duplicate. Well, now they're being pulled to opposite sides. So now both of my new cells are going to be identical. They got the exact same. They got the full set. Oops, sorry, that's my alarm. They got the full set of DNA, so they got like both of my 1s and both of my 2s and both of my 3s all the way through 23. But they went to opposite sides. So now they no longer have that double amount of DNA. They have what they need to be, identical to the original. So they get pulled to opposite ends. The reason when I drew this during first period, they said, why are they shaped like that? Well, I just told them that think about this as your elbow, And if somebody tied a string to your elbow and started pulling, your arm would kind of be like in an L shape. So these are just being pulled. So I just kind of made them do like that. And then the last phase is telophase and cytokinesis. So sometimes you'll see these separated, but cytokinesis is just the end of telophase. It's where the final two cells have been created. So the chromosomes reach their destination of opposite ends of the cell. The spindle fibers, kind of go away. The nucleus begins to reform, and the DNA or the chromosomes are able to relax, kind of start to unwind down into the loose DNA. DNA stays loose in the nucleus so that we can get to its information. If it stayed in chromosome form, it would be too tightly packed and would never be able to make, RNA or send out any of the signals that the cell needs. So it begins to loosen up in those nucleus as the cell membrane begins to pinch. So we've pulled that so far that it's starting to what's called create cleavage, which, it's the same thing. It's like just a crack. But the final splitting is like what now people are calling cytokinesis. It's just the end of telophase. And it's where you get two brand new daughter cells, and they are identical. They have all the loose DNA in there, and they have all 23 pair for humans. And we're back to just having 23 pair instead of that diploid number where we had, double the amount that we were supposed to have. And once this happens, both of these new cells will now, because that's telophase, they'll both enter G1. And they're independent living things, and they'll just, do their cell cycle.